Hello dear students. Today we will study a very interesting topic, and that topic is Human Genome Project, that is HGP. We will study the various aspects of the project. Let us find out what are the interesting features of this project. This project was officially launched in the year 1990 and it was officially concluded in 2003, or you can say it was a 13-year long project. Even after 2003, there were some official statements were given about this project in 2006. So you can see it was a very vast and elaborated project. That's why this project is called a mega project. There are various aspects of this project which are so interesting. It was coordinated by various agencies. Let's see. First was the US Department of Energy along with US National Institute of Health. They were also assisted by Wellcome Trust of UK. Apart from that, many other countries like Japan, France, Germany, China, and many other more countries, they also contributed some way or the other in completion of this project. Now let's go into the details of the project. This project is called a mega project because we see the magnanimity of the project. Let's find out. We know that in human cell or the complete human genome contains 3 into 10 raised to the power 9 base pairs. And if we see the cost of sequencing this genome, let's find out. If we consider, in fact, it was estimated that for sequencing one base pair, it would be costing around $3. So the approximate cost which was estimated was $9 million. It's a very costly project. Then these sequences, because there are 3 into 10 raised to the power 9 base pairs. So to uncode, to sequence them, if we consider them typing, suppose one page of any book contains 1000 letters and each book has got 1000 pages, then it was estimated that for sequencing the complete human genome, 3,300 such books would be required. So it would be a very massive project which was not possible manually. So for this project, different computational work was done. Supercomputers were formulated, they were prepared for it. Different software was built for this. It was made for this project. And a new branch of biology came up. And this branch of biology was called bioinformatics. So this bioinformatics was basically applying genetics and using computers to study the genetics. So it was an integration of biotechnology and information technology. That's why it was called as bioinformatics. So these were some aspects of this project. Now this project, because the complete sequencing had to be done, various approaches were used. There were two main methods which were used. First method was called EST, which stands for Expressed Sequence Tags. I will tell about it in more detail. And another was SA, Sequence annotation. What is EST? We have already studied that in case of eukaryotes, like humans are eukaryotes, we have the sequences, that is gene in the formation of, in the form of that is exons and introns. You know that exons, they express themselves, and introns, they do not express. So there is sequencing is done, and then splicing is done to separate the introns and exons. So in first approach, all the genes which were expressed as RNA, only those genes were first sequenced. So this was first approach. So all those non-coding sequences were not sequenced. Okay. Whereas in case of another approach, it was a blind approach where the complete DNA was sequenced. So there it was not segregated into introns and exons, or the parts which are expressing themselves, or which the parts which are not expressing themselves. It was a continuous expression. Okay, continuous sequencing. Now, how was this procedure done? For doing this procedure, first of all, the DNA was isolated from the human cell. After the DNA was isolated, because DNA is a long thread-like structure, this DNA was fragmented by using specific enzymes. And these fragments were later on amplified with the help of spatial vectors like BAC, bacterial artificial chromosome, and YAC, yeast artificial chromosome. When we will study about that gene cloning, we will be studying more details about these clones, these plasmids. Okay, so amplification of each piece was done by a specific vector, and now these fragments which were produced, which were obtained, they were sequenced, and this sequencing was not possible through just studying under the microscope. So for that, automated DNA sequencers were produced, they were formulated, they were made. So Frederick Sanger was one of the scientists who had done this automated sequencing in case of studying the amino acid sequences in proteins. So similar types of DNA, automated DNA sequencers were produced. Okay. And then these were studied for the sequencing. And the sequencing was done by studying the overlapping of different fragments, and in this way the complete sequence was made. Okay, so this was a general idea about the Human Genome Project procedure. We will be studying more about other aspects of the Human Genome Project through your book, where we will study about different salient features of Human Genome Project, and we will study various applications and challenges which were faced while studying the Human Genome Project. So let us study further what are the different aspects of salient features of Human Genome Project from your book. So before going into the details of the various goals and salient features of Human Genome Project, we must understand that this Human Genome Project, though it was very costly project, but it opened the research for so many other non-human models where the sequencing was done. For example, sequencing was done for various types of bacteria, yeast. Then there was a free living non-pathogenic nematode, Caenorhabditis elegans. Likewise, Drosophila fruit fly, its sequencing was also done. And many crops like rice and Arabidopsis and others have also been sequenced. So this opened up the research for so many other allied fields. Let us now switch over to Human Genome Project, its goal and the future other salient features and its application and challenges. I told you that Human Genome Project was a mega project. So what were the goals for this mega project? Let's find out. These are some of the very important goals of Human Genome Project. First goal was to identify all the approximately 20,000 to 25,000 genes which are present in human DNA. Then next, to determine the sequences of all 3 billion chemical base pairs that make up human DNA. Third goal was to store all this information in databases. Fourth, to improve the tools for data analysis because supercomputers were required and special type of software had to be developed for it. Next, fifth, to transfer the related technologies to other sectors such as industries. And sixth, very important, to address the ethical, legal and social issues, which is ELSI, that may arise from this project. So these were some of the very important goals which were taken into consideration for this Human Genome Project. Let us now study the salient features of human genome. Now, after studying HGP, many earlier assumptions were totally changed and very new concepts came into light. Let us see them. Number one, according to HGP, the human genome contains 3,164 million base pairs. Second, the average gene consists of 3,000 bases. It was observed. But it was also observed that the size may vary greatly. For example, the largest known gene, which is human gene, which is dystrophin, has 2.4 million bases. Third, the total number of genes is estimated at 30,000, which is very much less as compared to the previous estimate, which was around 80,000 to 1,40,000 genes. Earlier it was thought so. And almost all 99.9% nucleotide bases are exactly same in all humans. Fourth, the functions are unknown for over 50% of the discovered genes. We don't know. Still we don't know. Fifth point, less than 2% of the genome code for proteins. Others are mainly regulatory genes. Sixth, the repeated sequences make a very large portion of human genome. We will study it when we will study about the biotechnology. Then repeated sequences are stretches of DNA sequences that are repeated many times, 100 to 1000 times. They are thought to have no direct coding functions, but they shed light on chromosome structure, dynamics, and evolution. In fact, these repeated sequences are the basis for DNA fingerprints. They act as markers of the genes. Then next, eighth point, chromosome number 1 has most number of genes, which is 2,968 genes present on chromosome number 1, whereas Y chromosome has fewest number of genes, only 231. Ninth point, scientists have identified that about 1.4 million locations where just there is a single base differences, which are known as SNPs, single nucleotide polymorphism, which are pronounced as SNPs. This information promises to revolutionize the processes of finding the chromosomal location for human diseases which are associated with human history. Like many diseases are because of just a single nucleotide polymorphism. So, at the end, let us find out the future applications and future challenges related to Human Genome Project. So, with the application of this Human Genome Project, we can decode so many different aspects of human genetics. We can derive meaningful knowledge from the DNA sequences, and we can use it for various other public and private sector worldwide. Okay, so one of the greatest impact of having human genome sequence is basically enabling radically a new approach to biological research. In the past, researchers studied only one or few genes at a time, but with human genome sequencing or new high throughput technologies, we can approach questions systematically on a much larger scale. So, with the help of this Human Genome Project, we can understand how the different tissues work and how there is just an orchestra of so many different genes. They are interconnected to orchestrate the chemistry of life. So, this Human Genome Project is being used extensively nowadays in medical sciences, and in coming times it will have more prospects in understanding the human biology in a better way. So I hope you have understood the whole concept of Human Genome Project. Many questions are asked based on various aspects of Human Genome Project. So please understand and learn them properly. Thank you.